Day. It's 8:30 here. I just wanted to welcome you again for a celebration of science. And without any exception, we have a superstar today. I worked with Artur for many, many years and discovered incredible quality of his research and precision and everything wonderful about him as a scientist, as a colleague, as a friend. So Artur, we're more than delighted to have you today. And let's just switch to Dr. Aru.
Good morning, everyone. My name is Arun. I'm a postdoctoral fellow working with Dr. Paul Chesky. I'm pleased to introduce our guest speaker, Dr. Artur Cideciyan, a research professor of ophthalmology at the University of Pennsylvania. Dr. Cideciyan obtained his Master of Science and PhD in biomedical engineering in 1988 and 1992 at University of Miami. He studied research assistant professorship in 1993 at University of Miami. Then he studied faculty positions in ophthalmology at the Shanghai Institute in the School of Medicine at the University of Pennsylvania since 1995. Dr. Cideciyan has appointed as a co-director of the Center for Hereditary Retinal Degenerations at the University of Pennsylvania since 2010. Dr. Cideciyan was given various awards for his remarkable research, such as the Visionary Scientist Awards from the Hope for Vision in 2008, the Senior Scientific Investigator Award from the Research to Prevent Blindness in 2010, and the Proctor Award by the Association for Research in Vision and Ophthalmology in 2018. Dr. Cideciyan's research focuses on understanding disease mechanisms and evaluating treatments in hereditary retinal diseases using various research methods such as optical coherent tomography, electrogram to evaluate the rods and cones, and pupillary imaging to define post-retinal visual function. He has published over 200 peer-reviewed articles such as PNAS, Nature Medicine, and Human Molecular Genetics. Dr. Cideciyan also joined multiple clinical trials as an investigator since 1994. Very interestingly, his recent ongoing international clinical trial is to investigate a gene-based RNA therapy in patients diagnosed with Leber congenital amaurosis caused by CP290 mutations. Today, he will be speaking about improving visual function of foveal cones in inherited retinal diseases. Please give a welcome to Dr. Cideciyan.
Well, thank you very much. Thank you, Chris, and thank you, Grazina, for inviting me, and thank you for that generous introduction. As I was just mentioning a few seconds ago, Friday late mornings are really a pleasant time for me to be joining this seminar and learning from it. And I hope I can contribute a little bit to that. And today I'd like to tell the story of two diseases and two treatments and their consequences on the fovea a little bit, and two cases where I consider one of them probably a failure and the other one probably a success. And of course, that's my biased opinion, and I have no relevant disclosures. Our group has been very interested in inherited retinal diseases and their treatments, which inherited retinal diseases, of course, refer to a group of disorders caused by single gene defects that act in or on rods or cones or RPE cells. And, we now know that there is 300 or more genes that can be mutated, thousands of mutations in these genes, and we have learned a lot. But things were maybe a little less clear back when I was starting my career circa mid-1990s. So on that graph, we would be down here. It's not that we didn't know anything. We certainly did know quite a bit. In terms of gene mutations, we knew about rhodopsin mutations causing dominant RP, and we knew about PD6 beta mutations causing recessive RP, and RPGR mutations causing X-linked RP, et cetera. And we had decades of experience with these RP-like diseases, and there was a general thought, at least in my own simplistic mind, maybe others had a much more complicated thought process. And in my mind, RP was a natural history could be explained by such a simple graph. This was early in the disease. This is late in the disease, good and bad, and structure and function started good. And then at some point, things turned, took a turn to the worse. And both structure and function got worse. There was retinal degeneration and loss of visual function. And this shape of the curve could be approximated to a very rough level to a delayed exponential by some mechanisms. There were also other diseases, of course, such as rhodopsin kinase mutations and arrestin mutations causing forms of congenital stationary night blindness. And again, a very simplistic point of view of these stationary, quote unquote, stationary diseases was that the retinal structure more or less throughout the life of the person, of the patient, did not change or did not change much. There was a severe dysfunction from early on, and that severe dysfunction remained throughout the life, and there was a dissociation between structure and function. Why is it important to think about, the natural history of retinal diseases? It's because, we all wanted think about treatments, and of course, when one has a simple retinal degeneration natural history, one can think of a treatment that acts early in the disease that can help retain the function and structure at a normal level, or much more likely was those of you who are a little older will remember Elliott-Burson type of trials where the treatment would be acting later in the disease, and the aim would be to either stop or slow down further progress. There was less thought about stationary, congenital stationary type of diseases, but the presumption was that if there was a treatment, it would return function back to structure. So it would get rid of this dissociation and make function as good as the structure can be. In 1997, there was a what would later we would recognize as a seismic shift, really, in understanding and treating inherited retinal diseases. That was the year where it was discovered that our P65 mutations cause forms of very early onset disease. And the significance of that was not necessarily obvious to people like me, but people who are much smarter than I at the time of this publication recognized the significance, such as my friend Alan Wright wrote back in 1997 that our P cell is much more amenable than photoreceptor cells, both to gene therapy and transplantation approaches, and making them a good candidate for future therapeutic efforts. So that would be, that we would now know to be true 25 years later. Soon after 1997, there were also animal models. There was a naturally occurring animal model with Gasagiri and colleagues discovered with the RP65 mutation, and histology showed lots of photoreceptors in the eye, but when one did ERGs, there was very little signal. That was interestingly unexpected, one can think. Michael Redmond's RP65 knockout mice similarly showed on histology lots of nuclei, lots of photoreceptors, very little function. So the thought was, what does this this kind of disease natural history look like? It certainly didn't look like the simple retinal degenerations of progressive retinal degeneration and progressive loss of function, and looked more like the stationary night blindness type of diseases. And indeed, Gasagiri and colleagues initially called this dog as a form of stationary night blindness. Um, Again, at the time or soon thereafter, people, very smart people like my friend Chris Palczewski, thought of how can one ameliorate this RP65 deficiency, which at the time was very clearly known to be stopping the biochemical cycle, the visual cycle producing 11-cis retinal. And him and colleagues came up with the idea of an oral 9-cis retinoid. And our group was lucky enough to collaborate with them at the time. And we showed that within a matter of hours, really, of oral gavage of 9-cis retinal, there was a huge increase in signal. And we were, again, very lucky to be associated with Gus Aguirre and Bill Halsworth and colleagues in a different approach of treating RP65 deficiency in the dogs. That was gene augmentation therapy. And again, we were involved in doing some of the non-invasive work. And again, we showed that that there was this amazing improvement in visual function in a matter of days to weeks after the subretinal injections. So what it appeared like, if this was kind of the basic model of the natural history of these mice and dogs, and that there was a dissociation between function and structure, that whatever treatment one was using, whether it's 9-cis or gene augmentation therapy, was certainly improving function towards what could be the potential from the retinal structure. Of course, even though over the years we have been involved in studies from mice to chickens to pigs to dogs to primates, our really bread and butter and our really main interest is in humans. And of course, at the time and since then, we have always been very interested in understanding what is the relationship of the human disease to the animal models. And or specifically, is the human a larger version of a mouse or a dog? So we were very interested in looking at the retinal structure of these patients. Here's an RP65 patient at 10 years of age, and these are OCTs. Many of you are, I'm sure, familiar with, in this case, along the vertical meridian going through the fovea. And again, as many of you may be familiar with, is that photoreceptor nuclei are located in a hyposcattering layer in the OCT, shown here as the outer nuclear layer. And the RP65 patient is showing either missing this layer or having it thinned. So even within the first decade of life, there is quite severe degeneration, at least along this axis. And we were very interested at the time to see the spatial distribution of the disease. And in a normal eye, the spatial distribution of the outer nuclear layer, it looks something like that in a pseudocolor map, where it's the thickest at the fovea and thins with eccentricity, but thins less so in the superior direction than other directions. If one looks at a bunch of, I believe, 11 maps of RP65 patients from age 6 to age 23, the first 3 decades of life, one sees a common theme. And the common theme is that everything is in very cold colors, if you will, on this pseudocolor scale, meaning thin, as opposed to the warm colors in the normal. So there is extensive loss of nuclei and thus retinal degeneration in these patients. So this looks very different than the mice and the dogs, at least at the initial look. And of course, we were very interested in the function of these patients, and we looked at the retina-wide sensitivity, and these are maps across the whole retina. And in this unusual kind of scale that it covers 7 log units, normal is mapped to white or pink, and what one can see is RP65 patients are from yellow to black, so anywhere from 3 to 7 log units down. So a dramatic loss of function. So there is degeneration, there is dramatic loss of function, and what is then the simplistic, if you will, the natural history of the RP65 disease? And it clearly is not that. It clearly is not a lifetime of retained photoreceptors that are dysfunctioning. is it the other model, the retinal degeneration model or retinitis pigmentosa model of things going down together? Well, to be able to answer that question, we would have to know at such a point in this model, what is really the expected relationship between structure and function? And at the time we were doing this, which was early 2000s, this was really not known or thought about. So we had to kind of come up with a way of measuring structure and function relationship in disease. And one thing we thought about is that the quantum catch of photoreceptors in the outer segments, in this case, rod outer segments, and to a first approximation, we said the total amount of rhodopsin in the outer segments kind of relates to total amount of sensitivity that can originate from these photoreceptors. And that in turn is related to the length of the outer segments as well as the number of outer segments. And we used a data from Paul Sieving's lab that showed in P23H transgenic rats that when you had reduction in photoreceptors, in this case a 4-to-1 reduction, outer segments didn't remain in the retained photoreceptors. Outer segments didn't remain in the original height, but they shortened, and they approximately shortened to about the same proportion as the retinal degeneration. So if there was a 4-to-1 loss of photoreceptor cell bodies, there would be approximately 4-to-1 loss of outer segment lengths. So using that, we estimated that if one says the output of a normal retina is 100%, a 4-to-1 loss of degeneration would result in only 6% of outcome, or almost a log unit of loss. And using that, we define models of relationship between outer nuclear layer thickness and dark adapted sensitivity, both of which we could measure in patients and both of which we could measure at specific locations in patients. And so we went ahead and in our P65 patients, we went and measured the outer nuclear layer, and at the same location, we measured their sensitivity. And the dark symbols are the results. And I don't want to go too much into detail, except maybe the simplest thing to do is to look at about half the thickness of the outer nuclear layer. So if this is normal, about half the thickness would be about here, and regular RP patients would have only a little bit of loss of sensitivity, whereas our P65 patients were showing log units more loss of sensitivity. So clearly, our P65 patients had a degenerative phenotype and they had a dissociative phenotype. And so it was a kind of a new kind of a thought process, at least for me, maybe not for others, where it was a complex disease that both showed dissociation of function and structure as well as a progressive retinal degeneration. And why was that important to think about? And because, again, we were always thinking about treatments and how treatments could act. And indeed, if one starts thinking about this kind of natural history, treatments could have many potential outcomes. A good treatment could undo the dissociation and stop the degeneration, or it could undo and ameliorate the dysfunction but continue the degeneration, or it could stop the degeneration but not be able to undo the dysfunction, or absolutely not work at all, and natural history would just go along as it does. Or one can even think about a so-called treatment being bad for the retina, and then the natural history would get actually worse than what you would expect. So we were very lucky again, a few years later, to be involved in a human trial using Chris Balchevsky's 9-cis retinoid. And here are two of our patients in that trial showing before and after results in terms of retina-wide sensitivity. And in a matter of days, as has been put in publication, but turns out to be in a matter of hours, there was a dramatic increase in rod sensitivity in these patients upon ingestion of the 9-cis retinoid. And so what we could show then is that function was moving towards what would be expected from the structural potential of these retinas. However, there was no long-term data to determine what happens to the degeneration after that. We were also very lucky to be funded by the National Eye Institute to have our own gene augmentation therapy, and there we could show that in many patients it was possible to undo the dysfunction and make this complex degeneration that is both dysfunction and degeneration into a simple degeneration by adding a substantial amount, up to 4 log units of rod function, and thus ameliorate again this dysfunction component of this natural history. I don't want to go too much into detail at all, but we have then also published extensively on what happens on the long term, 3 to 5 years after the treatment, after successful treatment. And what we showed is unexpectedly that the rate of degeneration did not stop, and instead it continued even though there was a successful treatment of the dysfunction component. So summary so far, this P65 form of Leber congenitalosis is a complex disease with both degenerative and dysfunction components. Oral 9-cis-retinoid can ameliorate the rod dysfunction in a matter of hours to days. Gene augmentation therapy can ameliorate the rod dysfunction in a matter of days to months, but the rod degeneration continues unabated after gene therapy. Well, rod, well, how about the cones? Well, it turns out in the case of gene therapy, we could show that the cones improve also. And here are some of the results we published. These are 15 individual patients that were treated, and across a row is each patient's results at baseline variability versus postoperatively, what happens in the same eye. This is all in the same scale. And one can see that in the first set of group of results, which are the R's, represent the rod-mediated results. And there's a whole bunch of bars, which are multiple visits, showing to the right, in this case, many log units of improvement. And then in the same eyes, in the same patients, C's, which are cone-mediated results, also show bars to the right, except clearly they're smaller. When one looks at the statistics, one could show that there was substantial and significant results for rods, about 1.6 log units, so that's about that much on average, whereas for cones, it was more like 0.45 log units, so about that much. So both rods and cones, but rods greater improvement than cones. Well, how about then the elusive holy grail, if one may say so? Does cone improvement mean that there was an improvement in sensitivity of the foveal cones? And we were very interested in that, and others were, of course, also. And but before answering that question, the first question was, did P65 eyes have any room for improvement at the fovea? Was there a lack of quantum catch at the fovea? And the answer is yes. Here are a bunch of young patients' OCTs measured at the fovea. We can see that the nuclei were thinner than normal. Inner segments were shorter, outer segments were shorter. And when one related the outer nuclear layer thickness to the dark adapted sensitivity at the fovea, this is the normals. These are other patients showing what is expected from a simple retinal degenerative phenotype. And these are the P65 patients, which clearly showed a different phenotype. Again, here, if we look at it, a normal fovea, which is about here, could be associated with 2 to 3 log units of loss of sensitivity. So there was plenty of room for improvement. But when we looked at our results, we didn't see any. And here are 12 of the 15 patients where this kind of data in our gene therapy study, that this kind of data can be obtained. Each one of the gray areas is the whole visual field. And you can see the optic nerve and the fovea, which is a little cross put on there for perspective of what we are covering. And the dashed lines is where the gene therapy vector went as a subretinal injection. And the green squares are the areas which showed substantial and significant improvements in vision at multiple visits. And what is clear is that when one looks at all the plus signs, none of the plus signs are lit up by the green. So at least by this measure, that the fovea is not improving in sensitivity. Well, how about instead, maybe we are overthinking this and forget sensitivity, forget quantum catch. How about just simple, visual acuity? Isn't that measure foveal function? Sure. In a normal eye, at the foveola is where we get 20/20 vision. That little square that is shown here is the size of the lateral size of a 20/20 target, and it refers to what is visible in a normal eye. But in many of these patients, visual acuity was at the 2200 level. At the 2200 level, there is no guarantee that it's originating from the fovea, and there is really no guarantee that it's originating from cones. But nevertheless, there was a visual acuity controversy back many years ago. Some groups originally suggested that some of the patients were improving in visual acuity, and then later from the same groups, there was phase 3 results that suggested that there was no statistical improvements in terms of visual acuity. In our study, we showed that visual acuities were about 1 logmar, which is 2200 before and after therapy, and there was no statistical improvements in our eyes when looked at it as a group. However, we are not known to be lumpers, and as you know, there are the lumpers and the splitters, and lumpers try to lump everything together. We'd like to think of each individual patient as an individual that experiment, if you will, at these early trials, and we would like to consider them separately. So I'd like to give you just a glimpse of an idea of some individual results with the RP65 patients. I'm going to show you 3 short videos. In each case, this is the treated eye being shown. The little white circle is the location of the anatomical fovea. Optic nerve and blood vessels can be seen. The overlaid white boundaries are the area that we can see and measure sensitivity increases that are because of the gene therapy trial, but in these experiments, a letter is being shown, and the letter can be seen here. A letter is being shown, and the fundus is being viewed while the letter is being shown, and the patient identifies correctly the letter, and we see what part of the retina they're using while identifying the letter. So it's a fancy version of visual acuity, if you will. And here's the example: eye movements due to the nystagmus, and the patient is using the superior temporal part of the retina, which was treated, to look at the letter K and recognize and identify it. Here's another patient, again, nystagmus, and there is this parapapillary region of increased sensitivity, and that's exactly what the patient is using to look at the larger letter C and recognize and identify it. And here is a third patient who had 2 blebs placed and 2 areas of improvement in terms of visual sensitivity, and one of them is being used by the patient and not the fovea to look at and identify the results. The summary so far then is that gene therapy for RP65 can ameliorate rod dysfunction in a matter of days or months. Rod degeneration continues unabated. It can also ameliorate cone dysfunction, but apparently only in the extrafoveal retina and not in the foveal retina, at least not in our hand. So this should be about halfway to my talk, at which point I'd like to shift gears and ask the question, are there other retinal diseases with dysfunctional foveal cones? And the answer turns out to be yes. There is actually several. We have published on several of them in the literature. But what I like to talk about is one of the more common causes of Leber congenital amaurosis caused by CEP290 mutations. CEP290, thanks to work of Anand Swarup and his lab, and Tad Wenzel and his lab, we know that is located in the transition zone between inner and outer segments of both rods and cones. And indeed, we have shown that it's in foveal cones, primate foveal cones also. And it functions by some method in holding structurally this connecting cilium together, the transition zone of the connecting cilium together. We have, of course, been very interested in the human phenotype caused by CEP290 mutations. We have done some work with the RD16 mice and the NRL double mutant mice in an attempt to understand and eventually treat this unusual disease. I'd like to give just a little bit of the most salient features of this disease over the next few slides. Again, here's another slide looking at the OCT. As I mentioned before, this is a vertical profile along the vertical meridian going through the fovea, and the OCT looks like that. And what's highlighted, again, as I mentioned before, is the outer nuclear layer where both rod, and cone photoreceptor nuclei are. In human OCTs, we cannot differentiate between the rod and cone nuclei. However, we do know that at least in normal retinas, there is a very high density of cones right at the foveola where there are no rods. And then quickly, when one moves outwards, rods and cones become equal density, and then rods completely overtake, so that by the time one gets into the more perimacular and peripheral retina, almost all of the outer nuclear layer is rod nuclei. So using that information, we can look at the CEP290 LCA OCT, and here it's shown at the same scale. And what we can see is there is a bunch of outer nuclear layer thickness in the center, and once one moves out of the central macula, there is either no or a very thin outer nuclear layer. So we interpret this as having lost all rod photoreceptors. Indeed, in many of these patients, this happens in the first decade of life. So a very severe degenerative phenotype that wipes out all or most of the rods. And what's left is central macular cones. And that's the kind of the salient feature of this disease. However, that itself is not extremely unusual. Indeed, in a lab like ours, where we see retinitis pigmentosa patients, this happens quite common. This is a stage of retinitis pigmentosa disease where there is a central cone-only tunnel. And indeed, many of the CEP290 patients would look just like retinitis pigmentosa patients with a central cone-only tunnel. The question, though, is what is the function of these kinds of locations at the fovea? And when we have actually looked at it in a few severely affected CEP290 patients, and here are some results. Again, this is the quantum catch relationship that I mentioned before of photoreceptor structure against sensitivity loss. And with the gray symbols are other ciliopathies such as BBS1 or Usher2A, RPGR, and they follow what may be called as a simple retinal degeneration, whereas the CEP290 has a very large dysfunction component, so that as we did before, if we drop down from normal to about half normal outer nuclear layer, most patients show very little change in sensitivity, whereas CEP290 patients can show many log units of sensitivity loss. However, we were very interested in not just the fovea, but also the parafoveal locations, and specifically what was the relationship of this whole area to function, because we could measure the structure, but we didn't know what to expect. And it turns out it's much more difficult to do this in terms of quantum catch, because at those locations, normal retinas have rods mixed in and difficult to guess as to what would be the cone component. So instead, we took a different approach. My colleague Alex Moroka took an artificial intelligence paradigm and took advantage of RP patients who have the same kind of structure as CEP290 patients. And So here are the 20 RP patients that had the stage of disease that looked like CEP290, and they had co-mediated function only in their central maculas. And he set up a deep learning algorithm using random forest to relate the input features of retinal structure from the OCT at each location, shown by the yellow bars, to target variables of measured sensitivity values, which are shown here in this one example. And when one applies that to RP patients, how well does their deep learning algorithm perform? Here are 4 RP patients that are not part of the learning process, and they're progressively greater disease from left to right. And the red and gray lines are the artificial intelligence estimates, and the dark red symbols, connected symbols, are the actual measurements. And not surprisingly, the deep learning algorithm learns very quickly how to associate what kind of structure that produces what kind of function, and estimates that really well. So now we can use this kind of deep learning into CEP290 patients in whom perimetry was able to be performed, and estimate and ask, what is the treatment potential? Do the patients act like they can improve their sensitivity? Well, some patients, to our complete surprise, turns out that what is predicted and what is measured is very similar, and they have no treatment potential. They have no improvement potential. Other patients, such as this one, across the central macula, they have very large treatment potential of several log units between what is measured versus what is estimated from the retinal structure. And then yet other patients are somewhere in the intermediate, where some retinal locations may show very little treatment potentials, whereas other retinal locations may show greater treatment potential. So the summary so far then is that CEP290 LCA is even more complex, one can argue, than P65 formal LCA, with rods showing a severe degenerative natural history occurring typically within the first decade of life. Central cones, on the other hand, showing, I didn't show this data, very little degeneration and lasting really over many decades. And individual patients in specific retinal locations can have surprisingly varied treatment potential. So once we are powered with that information, the obvious question is, can the central dysfunction be treated? And the answer turns out to be yes, although it's a qualified yes. So for that, we go to the fact that about 70% of the mutations in CEP290 are a deep intronic mutation, and it causes a classic splicing defect. And back about 10 years ago, it was shown in vitro that antisense oligonucleotides can fix this splicing defect. And so a company called Procure Therapeutics produced an antisense oligonucleotide called cepofarsen, and designed for this specific mutation that would be designed as an intravitreal injection. In 2017, a phase 1/2 trial started, a multi-center trial. We were part of it, and there was a total of 11 patients enrolled in this trial. And when one looks at it as a whole at 12 months, it appears like quite interesting and positive results, the pooled analysis. And specifically in terms of visual acuity, there's more than 5 lines of improvement. In terms of FST numbers, FST stands for full field stimulus testing, basically light sensitivity in the dark. It shows almost a log unit improvement on average. However, what was clear is that there could be large differences in efficacy, if you will, between individuals, even though this was an example of a clinical trial of extreme genetic homogeneity. Remember, they were all sharing the same exact intronic mutation, not just the same gene mutation. So we have tried our hardest to share with the scientific community as much of the data as possible, and 2 of them just got published this week, about all these subjects and some of the some of their results. But the one that I'd like to concentrate on the remaining few minutes I have is the implications for the pharmacodynamics of this antisense oligonucleotide, its durability, if you will, in foveal cones. To be able to do that, we were lucky enough to have the last patient dosed, which was patient 11, and this patient was dosed and received only one dose, and followed by 15 months of careful observation. Thus, we could see what a bolus does over time, as opposed to the original plan, which was to inject every 3 months. The key result was obtained by light sensitivity, and here are two graphs that show that. The lighter colors are the treated eye, the darker colors are the untreated eye. Top is the red stimulus, bottom is the blue stimulus, and the time scale is times 0 is baseline. It's the average of 2 separate baseline visits, and we are looking at log units of change in light sensitivity in these plots. And what happens is quite consistently, the treated eyes rise to a peak improvement in a matter of 2 to 3 months, and they remain at this peak improvement for the next 15 months at least. And there is a slow downward slope to these improvements, if you will. So that was very surprising to us, certainly. We also wanted to look at the more typical things such as visual acuity. These are the visual acuity results where both eyes improve a little bit, although the treated eye improves more than the untreated eye. The untreated eye improvement is not unusual in trials where visual acuity keeps getting repeatedly tested. The patient gets better at test taking, so there is a little learning effect. But as we were recognizing what was happening at about 3 months, that there was this dramatic improvement in light sensitivity, and the patient was voicing that they can see things much better in darker environments, we thought of measuring low luminance visual acuity. Low luminance visual acuity, for those of you who are not familiar, refers to the fact of just reading an acuity chart, except with either 1 log unit or 2 log unit neutral density filter, and thus making the whole environment, the ambient light levels, 2 log units, 100-fold dimmer. And just as the patient was voicing, there was a dramatic asymmetry between the treated eye and the untreated eye when one put 2 log units of neutral density filter in front of the eye, which of course would make sense because simply the light sensitivity was improved for the treated eye, which was shown in the previous graph. So when we ignore the fact that we don't have baseline data, and we plot intraocular differences over time, what we see is that it seems to approximately fit the same idea as I mentioned before, which is rising to a peak at about 2 to 3 months and a slow decline after that, but remaining positive or improved for at least 15 months. We also had the opportunity to look at microperimetry, and these are now, instead of light sensitivity measurements across the whole eye, we are making light sensitivity measures at specific retinal locations. This is the untreated eye. Black symbols are at baseline. Light symbols are for the next 15 months. And clearly, untreated eye is just variability. Nothing is changing. In the treated eye, on the other hand, the baseline is over here. And from month 1 to month 15, there is a dramatic increase in sensitivity, in this case, almost a log unit and a half, as well as broadening, meaning expansion of the visual field. Indeed, the patient voiced that before treatment, they really never used their tiny, small tunnel vision for any kind of reading. However, after the treatment, they were using their vision, more for reading. So we wanted to understand better what that could mean in terms of the data that we were measuring. And we simulated what could be their reading ability at baseline. And we guesstimated that they could only see about a single word at the smallest font that they could see. What happened after treatment is their visual acuity improved, so they could see a smaller font, and their visual field improved, widened, so they could see more of it. So that's what we think is approximately what's happening in terms of their reading. Well, of course, all the measures I've said so far are very subjective. The patient presses a button, the patient says that they see something, so it's subjective. And we would love, in general, to have also support from objective measures. And one of the objective measures we have used back from 2 decades ago in the original gene therapy work in the dogs is pupillometry. We can use it in mice, you can use it in dogs, you can use it in humans. And for those of you who are not familiar, one looks at a dark adapted pupil, which is relatively large and shows a short flash of light, and the pupil constricts, and the constriction is plotted here downwards. Pupil diameter is large, gets smaller, and then recovers. This is 2 baseline values for this untreated eye and treated eye for this patient. And at month 3, again, there is a constriction and redilation. So qualitatively, they all kind of look the same. But when one expands and looks at the time scale, when one sees that in the untreated eye, the latency of the red symbols, which are at month 3 after treatment in the untreated eye, are no different than baseline. Whereas in the treated eye, there is about 100 or so millisecond acceleration of the pupillary response, which is exactly what we would expect if there was a increase in sensitivity of the fovea and parafoveal areas. The other aspect that we were very interested in is, are there structural changes in the treated eye? And to do that, we looked at the OCT again, and here we are using what we have called LRPs, longitudinal reflectivity profiles, that produce these OCTs, and they have peaks and troughs. And specifically, the one I like to talk about is this ISOS peak. So that's this peak here, and it's believed to be originating near the junction of the inner segment and the outer segment. And of course, not coincidentally, CEP290 is expressed near the junction of inner and outer segments. So we were very interested in that. And the relative intensity of this peak is plotted down here at multiple locations. Let me first show the foveal location. This is from time 0 to time 15 months. And in the treated eye, which are the dark symbols, and the untreated eye, which are the gray symbols. And what is obvious is starting at about 2 months or so and lasting to about 5 months or so, there is a peak, there is an increase in the intensity of this location. And that's with shown with the arrow. And we have done that at multiple locations in the nasal retina. There is the same peak. And in the temporal retina, which is shown again on the other side, and this does not occur in the untreated eye. So we think there is subjective and objective measures, multiple measures, almost a dozen to be exact, that appear to correlate with or support an idea that there is a rise at about over 2 to 3 months and a slow decline over 15 months, such that at 15 months time point, there is still definitely an improved light sensitivity and improved visual function. So then in conclusion, biallelic CEP290 mutations are a common cause of LCA form ciliopathy. There's a retained cones over a very wide window of ages. Again, I didn't show this, but over the first 4 decades of life. In a subset of patients, retained cones show major dysfunction, and thus allowing the potential for any kind of treatment, whether it's AONs or gene editing or whatever. we had the opportunity to look at the consequences of a single intravitreal bolus of cepofarsen, which can result in an incontrovertible, in my personal and biased opinion, biological effect in human foveal cones by both subjective and objective measures. And the peak effect is relatively slow at 2 to 3 months. And we'll go back to that in just a minute. And the durability is at least 15 months. And then treatment response really should take into account a individualized treatment potential of patients, and we should not be lumpers, but we should be splitters and consider each patient as an individual as to their treatment. I think I have more time, and in this time, I like to stop the data presentation. And since I have such wonderful people in the audience, and I like to speculate a little bit about the biological basis of these observations, at least the way I see it, and hope that some of you can correct me or add to it. I'm going to show 11 of these schematics, and they're all going to be the same. So I'd like to take a second to explain. This is the eye. That's the location of the fovea. These are the foveal cones. And what we are measuring is the visual function over time after treatment. And what I like to do is speculate as to what's happening in this kind of measurement that we are making. That is, at baseline, this is not zero function. There is definitely some function, some visual function that improves and then slowly declines. And while we're doing that, I'm going to speculate about what may be happening inside an individual foveal cone. This is the cone nucleus. This is the inner segment. And these are the mutant mRNAs in this intronic mutation. And then there is a subset of the mRNAs that are wild-type mRNAs, and the ratio in the human fovea is not known, but it is believed to be some ratio extrapolated from the experimental studies, which means that there is some synthesis of CEP290, and some of that ends up wherever its correct location is at the transition zone. And we think, at least to a first approximation, what we are measuring is related to the cone opsin molecules, which are shown here with the black little wiggles. And so we think that there is some association between any changes here and the outer segments. So we go ahead and inject the cepofarsen into the vitreous, and it arrives at the retina, and that's time 0. And then it, according to the literature, it very quickly reaches the retinal nucleus, and there is and starts doing its antisense job, and presumably relatively quickly, hours, days, there is an increase in the wild-type mRNA. And upon increase of the wild-type mRNA, again, relatively quickly, presumably, there is synthesis of new CEP290. What happens after that, I'm not sure, but since this is a speculation, thanks to my discussions with Ted Wenzel, he suggested that some of these multi-protein complexes may take some time to form. So we speculate that these increase in CEP290 synthesis slowly starts making its way into the transition zone. And as it adds to the transition zone, somehow its structure improves, whatever the definition of improvement is. And upon improvement of that structure, there is greater cone opsin molecules, which we see in terms of measurements in the patients as an increase in light sensitivity. And this continues, and there is greater structural improvement, greater opsin molecules, greater function, and in about 3 months or so, we have the peak vision peak improvement that can be achieved in these in this foveal cones, and it just so happens that the longevity of the sepofarsen in the retina is not known. Some of the early results suggest as little a half-life of as little as 2 to 3 months. Others suggest maybe longer. But for this speculation, I assume that by the 2 to 3 months, it has disappeared from the retina, and thus the mRNA goes back to its natural state of less wild-type molecules, and there is less protein synthesized, and then we see a slow slow degradation, if you will, of this complex. And as the complex slowly degrades, there is less and less opsin molecules and less function. So that is certainly one way of speculating. So I'll stop there and see if you have questions and thoughts, and I hope I didn't put everybody to sleep.
We have many people who would like to ask you a question. Maybe we start with Jocelyn. Jolie? Jocelyn, Jolie?
Sorry, I can't show my camera. And the variability in response, that was a wonderful talk, Art. I'm always in awe of your work.
I don't think we can hear you.
It's garbled.
All right, let's move to Philip Kaiser.
Dr. Cideciyan, really, really nice talk. I enjoyed it. So my question is about the oral 9-cis retinoid therapy. So, you know, this is something that's conceptually very appealing, and there's been more data in humans with the dominant acting RP65 mutation that it's effective there, and we've kind of continued to study it. It's very interesting. So I was kind of curious what your thoughts are about the clinical future of that approach based on kind of your experience with it.
Yeah, it's absolutely, it's unfortunate that a decade has passed, and maybe Chris has some insight into that, but it's definitely very promising. We have a lot of data in 3 patients that we included in the original clinical trial. Unfortunately, that was not included or allowed to be included in the publications. I think they clarify a lot of information, and what we had originally suggested is that at a minimum, 9-cis can be used as to probe RP65 patients as to what kind of improvement they could get. At a maximum, at the right lifetime dose, it could affect the whole retina in terms of an improvement, as opposed to the gene therapy, which is, at least in the current approach, it affects a small part of the retina because of the subretinal injection.
Connie.
Hello, great talk. Really enjoyed it. I had a question about the effect of LCA2 treatment on foveal and extrafoveal cones. You showed us a really nice diagram of where was the injection and where was the response, but were any of the injections covering the foveal cones?
Yeah, so it's a great question. In our case, off the top of my head, 3 out of the 15 or 4 out of the 15 did include the fovea. We started with covering the fovea. We realized that the risk to benefit ratio of the fovea in that condition is not so balanced. So we made judgments on an individualized basis of that risk to benefit ratio, because clearly, at least in some of the patients, there was a negative effects of the subretinal detachment at the fovea. And so so it's a great question. I can easily talk an hour about it. It's a difficult question. Definitely some patients of ours that had the fovea detached did not improve at the fovea, although they improved extrafoveal. Thus, on that patient, it was a success. We knew the material worked, the result worked. It just didn't work in the fovea. So that's my basis, as opposed to there were some cases where it didn't work in the fovea, but it didn't work anywhere else either. So those I'm kind of throwing out, if you will.
Yeah, so and just one quick follow-up then. So in terms of the fovea's susceptibility to, let's say, bad consequences from the bleb, do you think it's just from that, or do you think foveal cones might also have a different sensitivity to the treatment?
I do believe that the foveal cones are different than the rest of the cones. I don't have, that's not an area of expertise for me. I don't know the full literature. I've read some of it. Certainly when you, at a very simplistic level, you know, the foveal cones, as you know, look more like rods than cones. And whether that has anything to do with the biochemistry, I don't know. In the original work that I believe we did with Chris, we showed we were at an early point, we were very worried that that RPE under the fovea was different than the RPE away from the fovea. And at least from an P65 expression point of view, there was no difference. So I do think that foveal cones may be different than extrafoveal cones.
Okay, and I don't want to belabor this, but that could be something that you address with the oligonucleotide injection as well, right? That you had, if response to that treatment, because that's a vitreal injection, you can get rid of the confounder of the injection at the structure that you're studying. So just curious whether you think this idea that the foveal cones are different is also reflected in the response to that treatment.
It is, and I had a slide I didn't show. Among the quote unquote failures of the AON treatment is that it happens in the first 2 degrees. And when you go out to about 4 degrees, which is like a millimeter, then you are, even though there is by our measures, there is a great treatment potential, but there is no movement in sensitivity that we are measuring. So in that case, it's kind of the reverse, if you will, but again, supports the idea that foveal cones are acting differently. Now there is, whether it's the cones themselves, there are several other hypotheses we can have as to why parafoveal cones are different, including something as simple as retinal thickness. But let's probably not go there.
Okay, thank you very much.
Artur, the good news is Mike Chittum is with us, so he will give a final comments and maybe you have a question, maybe not, but he will have a final word. But before, we have many questions from the room here. So let's move to John.
Hello, Dr. Cideciyan. Thank you for this amazing talk. My question actually relates to the question you just received. I was wondering about the patients that were using their extrafoveal regions to view the letters. Is the treatment is the treatment only therapeutic to cones that are directly tied to the rods? And do the foveal cones continue to degenerate because the treatment only really seems to help the rods and the cones that are tied to the rods?
Those are good questions. Unfortunately, my answer is going to be very short. The first one, the rod-cone relationship, I don't believe that the extra foveal cones in P65 need rod photoreceptors to improve, but I don't have the data to prove that. In terms of the foveal degeneration, that's something we have specifically stayed out of in the published literature, and maybe one of these days we'll will opine on that. I have no results to report.
Thank you.
Marco.
Good morning, Dr. Cideciyan. It's Marco here, a postdoc from CTVR. I want to know, do we need a diseased retina, a degenerated retina, to have oligonucleotides getting into photoreceptor after intravitreal injection? Or this happens all the time? Also in
Maybe Mike can comment at the end. Of course, my experience is all with diseased retina. And my guess is, I mean, certainly the primate studies and mouse studies suggest that normal retinas definitely have antisense oligonucleotides that reach into the retina. Of course, it's an intravitreal injection, and there is definitely a slope to the expression of these antisense oligonucleotides from the inner retina to the outer retina. So by the time you get to the cones, you have less of it. But nevertheless, I think even in normal retinas, they get there, is my guess and my knowledge.
Sam?
Hi, Dr. Cideciyan. My name is Sam, and I'm a MD PhD student in Chris's lab. And I was wondering about patients, especially with early onset retinal degenerations, if their visual cortex is not primed to really receive the full benefit of the treatment. And if you've thought about like visual therapy or trying to stimulate reconnections within the visual cortex.
Yeah, it's a great question we have struggled with for a long time. And the most, I didn't touch at all today, but we recently published on the subgroup of patients in the phase 1 trial of the antisense oligonucleotide that were only light perception for vision, meaning very severe vision loss, and they received the antisense oligonucleotide and how they responded. And you can look that up, but the 2-second summary is that the patients who had congenitally never had spatial vision did not gain spatial vision, although one can argue the antisense oligonucleotide worked just fine because they gained light sensitivity. They could simply see dimmer lights, but they couldn't determine spatial vision. So if that turns out to be true, at least that's what our data shows, then one has to think about how one can open the cortex to new connections and new input, etc. And that is, well beyond my comfort level in terms of, the literature. And I do know, for example, there are studies in amblyopia where they are using some drugs to be able to extend the critical period in children from a few years to up to 8 years, etc. Whether it's working or not, I have no idea. I'm not an amblyopia person, but it's a great question that allows for a lot of pondering at many levels.
Let's move to Vice Chairman Vladimir and then Andrew Brown.
Hey, Artur, great to see you, and thank you for this fantastic talk. Really, so much exciting data. I have a comment and a question about the first half of your presentation, the RP65 gene therapy. So my comment is about the failure of the therapy to restore cone function. We know from our own studies that it's very difficult in mice, even if we keep them in the dark and we give them sustained supply with chromophore, to see even a marginal improvement in cone function, whereas rod function is relatively easy, as you showed, and you increased and others have done in the past to restore rod function. So in addition, we also have shown that restoring a supply of chromophore specifically from the Mueller cells is both necessary and sufficient for proper folding and targeting of conopsin and restoring of cone function. And we did this in CRLBP deficient mice, where we selectively restore the expression of CRLBP either in Mueller cells or in RP cells. And only when we restored in the Mueller cells did we see normal cones, both structurally and functionally. So my comment is that maybe it's worth thinking about what the RP65 deficiency does to the Mueller cell pathway, and whether then when you do the gene therapy, there's a way to restore also this additional chromophore supply. Because again, based on our data, it's really vital for the folding of conopsin and sustaining the rod structure and function in the long run. And then my question is, broadly speaking, about this difference between mice and humans in terms of rod degeneration in RP65 knockouts. And the specific question is, has anyone looked at whether human rhodopsin is mislocalized in RP65 patients? And we know again that in cause, that's what happens, and it's one of the reasons, probably the reason for the cone degeneration. And again, and more broadly speaking, do you have any comments on why human rods are different from mouse rods in terms of susceptibility to degeneration in the absence of chromophore?
Great comments, great questions. I'm not so sure I have answers, but the combined therapy to try to get to fovea to respond, I think is a wonderful idea, especially considering there is more and more now intravitreal approaches to gene therapy. It may not be the craziest idea to think of the subretinal gene augmentation that we know works, and add on a intravitreal Mueller cell cycle gene-based therapy of some sort that we know will get to the foveal cones, because that's the thinnest part of the retina, and do some kind of combination. Of course, by the time one has that all working out, hopefully, my hope is that all subretinal gene therapy will go away and that everything will become intravitreal. But who knows whether that's going to take another 25 years or just a few years. So we'll see.
Now I'll give you Dr. Brown.
Thank you, Dr. Cideciyan, for a really kind of thought-provoking talk. I have a question and then a comment relating to your last couple slides. You showed convincingly the pupillary light reflex, the sensitivity testing, low luminance sensitivity testing. And my question is with regards to flicker fusion, and if that was considered as an assay for cone function specifically.
Yeah, no, we didn't, and we don't use flicker fusion specifically, and for no good reason. But well, our patients, we typically they come and visit is 3 days long and lasts from 8 a.m. to 8 p.m. So we don't really have any free time to add another test, but in theory, why not? Yes.
There's so much abuse they can take. With regards to the proposal for what could be the mechanism of recovery after treatment in those last few slides that you showed, and also in the context of the AI models that were used to try and predict the probability of recovery. In these AI models, one can kind of look under the hood and see what are they looking at to predict that. And so, as I'm a clinician myself, and so in acute inflammatory diseases that affect only the outer retina, there's something called acute posterior placoid multifocal pigment epitheliopathy, AMPI for short. And I can, if everybody's okay with it, I'll just share a quick slide here that shows one of these cases. This is someone who is a young gal who had an upper respiratory infection and then developed this presentation in both of her eyes. The white areas are abnormal, the orange areas are normal. And then the OCT looks like this, where you can see the disorganization of the outer retina. And clinically, when we look at people and try to predict when they're going to recover, we look for the external limiting membrane. And then, of course, for some constitution of ellipsoid zone or ISOS, as you talked about. So I think that one might be able to take or glean some information from these acute phase diseases to try and predict whether chronic genetic inherited diseases could benefit from recovery. But I think that's clinically one of the most important things, is identifying who are these people that are going to benefit the most. So I'd be very curious to see if these models do identify the external limiting membrane, which, as it turns out, actually is in this kind of whole schmutzy area here. And actually, we do see that it comes back after 6 months over here. And you can see some places where there's no ellipsoid zone or ISOS recovery, but other places where it almost completely normalizes. Anyway, I'll stop sharing that, but that's just got me thinking a little bit about the recovery and the potential for recovery in these patients.
Yeah, good thinking. I think without a question, one of the differences between, so I showed that RP patients and CEP290 patients approximately have a very similar appearance. That similarity really falls apart at the outer segment level. And so it's a little tricky to make the interpretation, because often RP patients have much better fixation and you have much better OCT. And these LCA patients, takes us sometimes 3.5 hours to get a single foveal scan. So, so there is practical differences, but in the subset of patients in whom you are confident that you're doing a decent analysis and decent measurement, there does seem to be the outer segment area is kind of, at the lack of a better word, mushy or not well lined up. And that's probably the reason why they are, they don't have function. And however, that's not the only specification of prediction of how much sensitivity they could gain. And you're right, we should, and we haven't done, look into under the hood, as you put it, in terms of the deep learning methods, and try to understand whether we can gain something specific from those patients. We'll look into that.
Artur, we have a fantastic conversation, a lot of wonderful ideas and questions. Let me entertain you with very, very, very short 2 last questions before we go to Michael. Mike is just anxious to make a final word. So Rafał, and then Dominique, and then we'll go to Mike.
Hi, my name is Rafał. I'm a postdoc in Chris Palczewski lab. I wanted to ask you about the heterogeneity of effects of your CEP290 trials. Is it related to age, or some other factors are involved in this? Can you comment on that, please?
Yeah, it's a good question. So age is not. In the first publication we published in 2019. The so-called hyper-responder was the oldest patient. It was a 44-year-old. Now, in retrospect, we have learned a lot since then. And in retrospect, we know that he was the patient who had no spatial vision at age 44 that we treated him, and neither did he have any spatial vision for the previous 10 years that we had followed him. But as a child, he had spatial vision and about 2200 vision. So we believe the reason he was quote unquote a hyper responder was unrelated to his age, was the fact that he had the cortical connections at an early age. He had the neurodevelopmental history to allow spatial vision once the light sensitivity went up at the photoreceptors. And whereas others who from early on did not have the neurodevelopmental history of having any spatial vision to the extent that we know, also increased light sensitivity, but did not increase in terms of spatial vision or obtain any spatial vision. They lacked spatial vision, and they lacked before, and they lacked after the treatment. So, so age, I don't believe, is very important, except for the fact that indirectly, not biologically, but in terms of catching, the patient that is in your clinic, the patient, the 44-year-old was there because we had followed him for a long time, as opposed to a patient, the more typical Leber congenital patients, which are typically very young because they have severe visual disability and they end up in the clinics. So there may be indirect effects of age in terms of who ends up in a clinical trial. But biologically, I don't believe there is a big difference from age 8 to age 40. Below age 8, there is much speculation that you could have some cortical effects, some who knows. So that remains to be seen.
Dominic.
Hi, Dr. Cideciyan. Quick question. So, if I understood correctly, in patients with affected CEP290, do the rods degenerate fast? So, is there any? What would be the difference in ciliogenesis or in cilia, basically, that keep the structure of cones unaffected, but the rods basically degenerate completely? What do you have any ideas?
No, it's a great question, and it really goes against the the so-called rod-derived cone viability and this and that. I mean, these cones, survive for decades, and there is no rod to be found. So in one sense, why are the rods being affected much differently than cones? And in another sense, once that defect occurs early in life, why do the cones survive after that? And I don't have answers to any of those, any of that question. And this is not just foveal cones, but so one could argue, foveola is happy with no rods in a normal retina anyway. But this central area extends to about 8 degrees or so, which is normally well into the rods and the mixture of rods and cones. So somehow rods drop out, we think, and cones survive.
And ladies and gentlemen, now I give you highly anticipated final words and wisdom from Mike.
Thank you, Chris. I should have left my camera off, shouldn't I? But it's a pleasure to listen to every time. It's an amazing series of work, incredible. The detail and the depth of the phenotyping that you do, Art, makes it possible. The length of the studies as well, the depth of the knowledge is what's really required to try and understand what's happening in the patients and how we can sort of try and bridge these gaps and hopefully move more therapies into the clinic and more effective therapies. And I look forward to seeing you at ARVO, hopefully, and we can maybe then talk about the interim results of the Sepafarsa. And
thank you very much. Thank you very much, Mike. Thank you, Artur. This was excellent. And what do we do next Friday? The same way, the same time, there'll be something.